Hi, I am Mariam Ram of TNQ. Welcome to the first of the TNQ-Janelia India COVID-19 Seminars. Today we feature Professor Susan Weiss of the University of Pennsylvania, and her topic is Coronavirus, Old and New. The COVID-19 pandemic has shut down much of life as we knew it, but science has risen to the challenge. In the midst of all the chaos and economic disruption, we believe it is important to present the science of the virus, SARS-CoV-2, the disease COVID-19, and the development of diagnostics, therapeutics, and vaccines. In presenting these seminars, TNQ is fortunate to partner with the Howard Hughes Medical Institute's Janelia Research Campus in Ashburn, Virginia, and to have the support of NCBS, the National Center of the Biological Sciences in Bangalore. TNQ conducts an annual lecture series in the life sciences. These lectures attract thousands who pack auditoriums and centers of scientific learning in India. Our aim has been to showcase renowned scientists in their current research and to have them interact with students, researchers, and faculty in the expectation that these interactions will inspire Indian students of science to pursue excellence and to do significant and creative work. These online COVID-19 seminars have the same objective in a very different, physically distant world. I will now hand over to Ron Vale, Executive Director of Janelia Research Campus.
Wonderful. Well, thank you, Mariam. And I also want to thank you for all of the inspiring work that you've done over more than a decade with the TNQ Distinguished Lecture Series. I know it's always a highlight of the life sciences in India. And this year, it's a great pleasure to work with you on behalf of Janelia Research Campus in co-organizing these virtual series on this extremely important topic of COVID-19. And we hope that it will fulfill a very important role in science communication on this subject in India and throughout the world. So I would also like to acknowledge the individuals from Janelia who are the co-organizers and have put in a lot of work into this series. Sarada Viswanathan and Janine Stevens. You will hear from them later as they will be hosting the question and answer session. And now I would like to introduce and turn things over to Professor Jitu Mayer, who is the director of the National Center for Biological Sciences in Bangalore. Jitu.
Thanks, Ron, for that. And thank you, Mariam. At a time when the COVID-19 crisis rages and India appears to be headed to becoming perhaps the epicenter of these infections, there has been a tremendous convergence of scientists the world over to provide possible avenues to address this crisis. There has also been an unprecedented collaboration among scientists near and far to understand the biology of the virus, study its mechanisms of spread, and of course, its amelioration. And true to its tradition of bringing the latest in cutting-edge science to Indian audiences, TNQ, this time in partnership with Janelia, brings to us some of the latest in scientific research that addresses these issues. And the National Center for Biological Sciences is, of course, delighted to support this effort. Thank you, Mariam, for doing this. For this talk, we have invited Susan Weiss, a veritable legend in the field of virology, and perhaps one of the world's foremost authorities on coronavirus, to provide us some food for thought and discussion this evening in India, morning in the United States. After the lecture, we'll have a panel discussion, following which there will be a questions from our audience. But before we get to the talk, let me introduce the panel for today. We have Dr. Shahid Jameel from Ashoka University and myself, and the discussion is moderated by Professor Vidita Vaidya. A few words about each of them. Dr. Vaidya is a highly decorated and outstanding neuroscientist and a professor on the Faculty of Biological Sciences at the Tata Institute for Fundamental Research in Bombay, where she has been since 2000. Her group focuses on understanding the neurocircuitry of emotion and those that underlie complex psychiatric disorders like depression and anxiety, very much common in these times. In addition to her research, Vidita is passionate about public engagement of science and scientists, and she has involved herself in many activities that keep alive healthy debates around science and its societal engagement, including issues around gender equality and access in science and science institutions in India. My fellow panelist is Dr. Shahid Jameel. Shahid began his career in science as a chemist and as a biochemist, and then he became a virologist, carrying out this work at the University of Colorado Health Sciences Center in Denver. And then he returned to India to set up the virology group at the International Center for Genetic Engineering and Biotechnology in Delhi, where his research focused, in fact, on human viruses. In particular, he's well known for his work on the hepatitis E virus. From 2013 to 2020, he served as the CEO of perhaps the foremost biomedical charity in India, the DBT Wellcome Trust India Alliance. And now he's the founding director of the Trivedi School of Biosciences at the Ashoka University. Shahid has been one of the most sought after scientists in India at this time with his virologist hat and his writings in the popular press. He has authored some of the most clear-headed pieces on the subject of today's pandemic in recent times in the Indian context. And now let me invite Sharda, who herself is a virologist, but is now engaged in science outreach and actively engages in training and educational efforts within the community at the Janelia Research Institute. Let me invite her to introduce Susan Weiss.
Thank you, Professor Meyer. Hi, everyone, and welcome to the TNQ-Janelia India COVID-19 Seminars. Let me give a quick overview of the order of events today. We will start with a talk from our speaker, Professor Susan Weiss, followed by a panel discussion where the speaker will be joined by Professor Meyer, Dr. Jameel, and Dr. Vidita Vaidya as a moderator. We will then move on to a Q&A session. Please note that questions can be written at any time during the talk or the discussion into the Q&A box on your YouTube or Zoom, wherever you're joining us from today. And we'll really appreciate if you could please indicate who your question is directed to, the speaker or the panelists. And now it's an absolute pleasure to welcome Professor Susan Weiss to the first of the TNQ-Janelia India COVID-19 Seminars. Professor Weiss obtained her PhD in microbiology from Harvard University, working on paramyxoviruses, following which she did a postdoctoral training in retroviruses at University of California, San Francisco. She's currently a professor and the vice chair of the Department of Microbiology and a co-director of the Penn State Center for Research on Coronaviruses and Other Emerging Pathogens at the Perlman School of Medicine at the University of Pennsylvania. A brief introduction into her work. Over the last 40 years, Professor Weiss has worked on many aspects of coronavirus replication and pathogenesis, making some fundamental contributions to the understanding of the basic biology, as well as organ tropism and virulence. She's worked on a range of coronaviruses, namely murine coronaviruses or MHV, MERS, and most recently the SARS-CoV-2. In today's talk, Professor Weiss will present a short history of coronaviruses, a summary of the coronavirus life cycle, followed by a description of the virus-host interactions. Professor Weiss, welcome, and we look forward to your talk.
Thank you so much for inviting me. It's really, really a pleasure to visit India, even though it's only virtual. I wish I could be there in person. Okay, so my talk's going to be entitled Coronaviruses, Old and New, and it's going to be divided into two parts. The first part is going to be a discussion of the basic biology of coronaviruses. And I think that's really important because it allows us to identify steps in the virus life cycle that may serve as targets for antivirals for many, if not all, coronaviruses. And I'll get to that more specifically. The second part of the talk is going to be on the incredible or the ability of coronaviruses to antagonize the host innate immune response. And this is really important because it's become become obvious recently that an inadequate or dysregulated innate immune response, or interferon responses in particular, have been associated with more severe disease, both in humans and in animal models. So coronaviruses are a subgroup of the nidovirus order of viruses. Nidoviruses are named for their nested subgenomic mRNAs generated during infection, nido meaning nest. They're envelope viruses, kind of medium-sized, and they have single-stranded positive sense RNA genomes. And these are the different groups of viruses that are all within the nidovirus order. Here are the coronaviruses. And just to highlight the coronaviruses, here's an electron microscope picture showing the characteristic corona-like morphology given the name. And these are the spike proteins protruding from the membrane that everyone's been hearing about, the spike proteins. This is a coronavirus particle, a schematic. There's a very long genome RNA shown here, about 30 kilobases. As I said, single-stranded, non-segmented. It has a capped 5' end and a polyadenylated 3' end as other positive sense mRNAs. It's complex with a nucleocapsid protein in this kind of helical conformation, and that's inside of a membrane that's derived from the host cell. And in the viral membrane, there are 3 proteins. The spike protein, shown here. The spike protein is really important in that it mediates entry, it binds to receptor, and mediates cell viral fusion. And I'll talk about that in more detail. It's also an important determinant of tropism, immune response, and virulence. The membrane protein M and the small membrane protein E are both transmembrane proteins also inserted in the membrane, and they're important for viral assembly as well as having some other intracellular activities. Some coronaviruses also encode a hemagglutinin esterase, shown here. That's not found in SARS-CoV-2. It's kind of a homologue of influenza hemagglutinin. But again, it's not seen but it is in some human coronaviruses, but not in the SARS-like viruses. I want to discuss the coronavirus timeline because, I mean, many people believe that coronaviruses just arose recently or have been studied recently, but actually they've been around for a really long time. And the earliest evidence of them in the scientific literature is in the 60s and 70s. So since the 60s, since that time, people have been studying the mouse hepatitis virus as a model coronavirus, many animal coronaviruses that cause severe diseases in animals, and there's been quite a lot of vaccine research on these viruses. In fact, probably the earliest virus in the literature is the infectious bronchitis virus, chicken virus. And then there were human cold viruses that were described at that time. Most known are OC43 and 229E. These viruses cause the common cold, but OC43 can occasionally infect the lower respiratory tract. So during the 80s and 90s, there was a lot of really basic coronavirus biology research, and I'll talk about what we know in a little bit, but it's because there was a sort of small group of people studying these viruses for all these years, so that when SARS arose in 2002, the first SARS-CoV, or SARS-1, we really knew a lot about coronaviruses. And it was because of that it was quickly identified as one of these viruses. It was sequenced, cloned, and everything was done rather quickly because of all the information that we knew before that. And that sort of signaled the beginning of the pathogenic human coronavirus era. And in the wake of the SARS epidemic, which only lasted 7 months, and we learned that SARS originated from bats and was transmitted through civets into humans. And after the SARS epidemic, people looked for other coronaviruses and identified HKU1 and L63 as human viruses causing pneumonia and bronchiolitis or croup respectively. And the other thing that was really important that happened after the SARS epidemic was that we realized that coronaviruses were quite common in bat species. And it turns out there are many, many coronaviruses in many different bat species, and that people are looking still very carefully at that. And then things were kind of quiet until about 2012 when MERS coronavirus arose in Saudi Arabia. And MERS came also from bats, was transmitted through camels where it has a reservoir, and people continue to develop MERS coronavirus, but at a much slower rate. It's less contagious than SARS-1 or SARS-2, but it's still around. And actually MERS is even more lethal than SARS. About 35% of people that get MERS die from it. Of course, we all know in 2019, late in 2019, SARS-CoV-2 arose in Wuhan, China. So SARS-CoV-1, MERS-CoV, and SARS-CoV-2 all cause severe acute respiratory disease. And just to be precise about it, COVID-19 is the name of the disease, not the virus. So SARS-CoV-2 causes COVID-19, just like HIV causes AIDS. And one other thing about the timeline, the first international coronavirus meeting met in Würzburg, Germany in 1980. There were about 60 people attending. I had just opened my lab as an assistant professor at Penn, and I was at that meeting. The 10th meeting happened in the Netherlands in 2003, just after SARS, and this one attracted hundreds and hundreds of people because of the SARS epidemic. And then the 15th meeting was supposed to occur this May in the same place in the Netherlands, which was kind of eerie, but it was postponed due to the epidemic. So I just want to discuss a little bit about the coronavirus genome structure. So the human viruses are found among the alpha coronaviruses and beta coronaviruses. The alpha coronaviruses include 229E and NL63. These are the human viruses. The beta coronaviruses have three lineages, ABC, now called MBECO, SARBECO, and MERBECO. And in the alpha, the lineage A viruses include HKU1 and OC43, and the murine hepatitis virus. I'm just listing it here because I'm going to talk about it later. The beta coronaviruses include SARS and other SARS-like viruses found in bats, and the lineage C viruses, MERS, and other MERS-like viruses. So as soon after SARS-2 arose, it was sequenced and was shown to have a very similar genome structure and sequenced to SARS-1, and hence it was labeled SARS-2. So all the lineages of coronaviruses, including the animal viruses, have a very similar genome structure. This very long replicase gene that's divided into ORF1A and ORF1B, open reading frame 1A and 1B, which encodes 16 non-structural proteins that make up the replicase. The spike protein is found next to that, and then the other structural proteins are encoded in the same order, always spike, E, M, and N for all of the viruses. What's different among the genome structure is the 3' N, or the accessory protein shown here. These numbered open reading frames all encode non-structural accessory proteins, and these are often the agents of interferon antagonism that I'll talk about later. I just wanted to make this statement before I go on, because there's been a lot of rumor and scuttlebutt about whether this virus could be engineered by humans. I'm quite sure that it's not, for really several reasons. Very similar viruses are found in bats, even viruses with spike proteins that would be potentially able to infect humans directly without an intermediate species. It doesn't resemble any known recombinant viruses, so it really wouldn't be easy to put together a virus like that. And finally, I think the most compelling thing is that it really seems impossible that anyone would know how to design a virus with the properties of SARS-CoV-2. So I'm completely convinced that this virus is a natural occurring virus that came from bats, most likely. Okay, the model of human coronavirus replication. This is a very simple schematic that I drew actually after the first SARS epidemic, but it makes the point. So the virus spike protein attaches to its receptor. It's then enters the cell by a virus host fusion event, and the viral nucleocapsid is released into the cytoplasm, where it's translated into a replicase protein. The replicase, or complex of proteins, synthesizes new genome RNA and also a whole set of subgenomic mRNAs, and I'll talk about the mechanism more. These RNAs are translated into the proteins. The membrane-bound proteins insert into the intracellular membrane shown here, MN and spike. The nucleocapsid combines with the RNA, and the nucleocapsids then bud into intracellular membranes at the intermediate compartment between the endoplasmic reticulum and Golgi shown here. And then virus particles are visualized in vacuoles, and they're transported to the cell membrane where they're kind of released into the medium. The spike protein is also deposited on the plasma membrane, where it can cause fusion among cells, and SARS-CoV-2 does this quite well. So I'm going to highlight now some things about viral entry, then genome mRNA synthesis, and then finally translation of the conserved replicase proteins that I mentioned before. So coronaviruses enter the cell by two routes, and let me explain those routes. The first one is the direct plasma membrane route. By this route, the viral spike protein recognizes its receptor shown here, which is, in the case of SARS, is angiotensin converting enzyme. a cellular or membrane-associated protease then cleaves the spike protein, and that activates it to mediate a fusion between the viral and host membranes, and the nucleocapsid is released into the cytoplasm. By the other route, the endosomal route, it starts the same way by the spike protein recognizing its receptor. The particle is then endocytosed into the cell, into an endosome, in which location the low pH environment of the endosome triggers this cathepsin, another protease, to cleave the spike protein, and again activate it to produce or induce a viral host fusion event, and the nucleocapsid again is released into the cytoplasm. So which of these routes is used? Well, this depends on a lot of factors. It depends on the proteases available on the host cell membrane, within the host cell endosome, and also within the host cell. The Golgi often contain an enzyme called furin that will also cleave the viral spike protein and actually sort of pre-activate it on the way that during its assembly, so that when it's produced by a cell, it's already pre-activated for fusion. So I'm going to give an example. So this depends on these proteases, but also on the spike protein. So let me talk a little bit about the spike protein and give an example of how we know, or how it's decided which route is used. So this is from mouse hepatitis virus. So all coronavirus spike proteins are divided into two subunits, S1 and S2. And this protein has to be cleaved in two places to activate its fusion. One at the S1, S2 boundary shown here, and one at S2 prime here. The cleavage here reveals a hydrophobic region that serves as a kind of fusion peptide that actually inserts into the host cell membrane to mediate fusion. So this cleavage site here is sometimes a furin, like a furin site, a site for digestion by that intracellular furin enzyme, which is really just a run of basic amino acids. So a virus like JHM is one of the strains of MHV, has quite a basic furin site, and it's cleaved during its assembly, shown here, so that it comes out of the cell partially activated, and then it enters the cell by this direct plasma membrane route after being cleaved by a plasma membrane-associated protease like TMPRSS2. The MHV2 strain, in contrast, closely related strain, does not have this basic furin site. It's not cleaved, and it enters the cell by the endosomal route, where it gets cleaved by cathepsin at both cleavage sites. The A59 strain is partially cleaved. It has a less basic site here at S1, S2, and it enters the cell by both pathways. So and all of these viruses, I should mention, are completely pathogenic. So why is this important? Because I think it's important because when we think about viral inhibitors, or we think about which pathway, we need to think about which pathway is being targeted, because chloroquine, for example, or cathepsin inhibitor, E64, will target only this pathway. And that's probably one of the reasons that chloroquine doesn't work very well. But a protease inhibitor like Camostat, which inhibits TMPRSS2, might inhibit by this pathway. But many coronaviruses can use both pathways, and in fact may use different pathways in different cell types. So it really is important to understand how the virus is entering the cell before designing antivirals that target viral entry. And now I want to talk about transcription of coronavirus mRNAs. So here's the genome RNA. It's got a 5' capped end, a 3' polyadenylated end, and there's short, about 100 nucleotide leader sequence at the 5' end of the genome. So to initiate transcription or RNA synthesis, an RNA-dependent RNA polymerase, or RDRP, is translated from the genome. It then transcribes RNA from the 3' end of the plus strand to produce a negative sense antigenome, which is then copied back into more plus strand genome. So this is how the genome RNA is replicated. At the same time, the polymerase or the replicase generates a set of subgenomic negative strand RNA shown here. And this happens by a really interesting non-contiguous transcription process, whereby the polymerase transcribes from the 3' end of the genome, and when it gets to this transcriptional regulation sequence, it jumps all the way to the 5' end of the genome, where there's a similar sequence, and continues transcribing here, putting a leader or anti-leader sequence on the end of the RNA. That's shown here. And then at some frequency, it skips this first transcriptional regulation sequence, goes to the next one, and then the polymerase with its nascent chain jumps to the end here and puts on the leader, making the second RNA. And that goes on until all these RNAs are made. And then the replicase transcribes back the plus strand mRNA. So the blue are the messenger RNAs for all the proteins. This is kind of complicated scheme. And so what do we learn? So one of the implications of this scheme is on the RT-qPCR tests that are used to detect coronavirus RNA. So here's an old gel from my lab of the RNAs of mouse hepatitis virus, but SARS would be very similar. We see a lot of the smallest mRNAs, less and less of the larger ones, and here's the genome RNA. So many of these tests for coronavirus genomes are actually qPCRs within the replicase gene, or the RDRP shown here. So this would amplify genome only. But other tests use the nucleocapsid primers, which would amplify here, which would amplify all of the RNAs. And this is actually a much more sensitive test for virus than this one, but this one detects replicating virus as well as genome virus. And I'm still kind of confused, and I'm not sure whether we think that during testing for SARS-CoV-2 in the nose or nasal tests, whether we're actually looking at genome or messenger RNAs or replicating RNAs. But this is just something to think about when we think about testing and what it means. Okay, translation of coronavirus proteins. So here are all the RNAs that I described before. Each one of them is translated from its 5' end, usually into one protein, but occasionally into 2 proteins shown here. But what's really interesting and more complicated is translation from the 5' end, this 2/3 of the genome. It's about 20 kilobases of RNA. So RNA translation initiates at the 5' end and translates here until the end of ORF1A and produces a protein called PP1A. And there's a termination codon here. But there's also a frameshift sequence at the end of PP1A that's shown here. It's a pseudoknot seen in the RNA, and it causes the ribosome to kind of stall a bit and slip back 1 frame, so that translation it goes back 1 frame and then continues through to synthesize the rest of ORF1B to get this really large polyprotein called PP1A1B. And while this is occurring, this is being processed into smaller proteins by the viral encoded proteases, which I'll talk about in a minute. So coronavirus can serve proteases and replicase. So here's again a diagram of open reading frame 1A and 1B. There are 16 non-structural proteins encoded. NSP3 encodes a papain-like protease, which cleaves at these 2 sites here. This is during translation. The 3C-like protease, or the main protease, processes at the rest of these sites, again during translation. And then the RNA polymerase is encoded in NSP12 and is actually the part of the replicase gene that's translated at a lower frequency. So ORF1B is translated approximately 20% of the time compared to ORF1A via this frame shift. So this is true of most viruses. They seem to make less polymerase than the upstream or the protease part of the genome. And this is the target of remdesivir that everyone's heard about, and remdesivir will work against probably all the coronavirus polymerases. These proteases have been used against, these protease inhibitors have been used against 3CL-Pro. I don't think they work very well. So in summary here, the proteases process replicase proteins, and the RDRP replicates genome and transcribes mRNA. I'm just noting these guys here because all of them have been working on antivirals to coronaviruses even before the SARS-CoV-2 emerged. There are also a whole bunch of other proteins encoded in ORF1A and 1B. Many of them, they either have functions in RNA synthesis. For example, there are this helicase, this activator and primase. The others are, and this proofreading enzyme that keeps the coronavirus from accumulating too many mutations and becoming too attenuated. There are 3 enzymes involved in capping of the 5' end, shown here, NSP16, 14, and NSP13. This endo-U is really important for antagonizing host immune response, and actually the capping also protects the mRNAs from being recognized by the host cell immune responses. So and then in addition, NSP1 and NSP3 encode further host antagonist activities. This one knocks down host translation, and this one has a deubiquitinating enzyme and a macro domain. So in summary, these enzymes promote synthesis and stability of viral RNAs, capping of 5' ends, and really a lot of protection from host cell sensors and interferon response. And they're all quite conserved. So any one or many of these proteins could potentially serve as potential drug targets. And here's an example of something I collaborated with Stefan Serafianos way back, where we looked at the inhibitor of the helicase. So here's the NSP13 of the SARS. This is the first SARS virus, a model of its helicase, this was actually a real structure, and predicted structures for the MERS and MHV enzymes. And this compound here, SSYA, fits into the binding pocket, theoretically, of all three of these enzymes. And it also inhibits replication of all of these viruses. Shown here is the replication of virus as a function of increasing concentration of inhibitor. And it's quite effective against all three of them. So there are many other proteins and other examples of how this could work. So this is just one example. This is how remdesivir would work as well. Okay, so in summary of this part, there are many conserved features of coronaviruses: the subgenomic mRNAs, the non-continuous RNA synthesis, the ORF1A, 1B frame shifting, all of these possible things that can be interfered with. There are proteases, there are RNA-dependent RNA polymerase and RNA modifying enzymes, and further host antagonist activities. And again, all of these are conserved among all of the coronaviruses and may serve as potential targets for antivirals, which I think is really important since we never know what the next emerging virus is going to be like. So now I'm going to flip to talking about antagonism of the double-stranded RNA-induced antiviral pathways by coronaviruses. And just the first slide is just going to show how well coronaviruses are at evading or antagonizing innate immune responses, in this case, interferon lambda and interferon beta. So here's MERS coronavirus as a function of time after infection. We see that at most there's a 10-fold increase, and this is just mRNA by qPCR in A549 cells, that there's about a 10-fold increase in mRNA shown here for lambda and even less for beta. But if we look with Synbis or Sendai, two other RNA viruses, and here we're looking at a log scale, they induce up to 100,000-fold more of interferon lambda, for example. And this is just one of the interferon stimulated genes. So this really is, the induction is dwarfs what goes on with MERS. So I'm going to talk about some of the pathways that we study, not only interferon induction, and also summarize some of what we know from other coronaviruses about antagonism of these pathways, and then talk about our most recent work on SARS-CoV-2. So these are sort of known antagonists of MHV and also OC43, the human virus in the same class. This NS2 protein, which I'll summarize in a moment, MERS coronaviruses encodes these two, NS4A and NS4B, and actually SARS-1 encodes a protein called ORF6. And those are really probably the best defined of the antagonists among the other formerly known coronaviruses. So these are the pathways we study. They're coronavirus double-stranded RNA-induced antiviral pathways. Really, any virus will induce one or more of these pathways. So the coronaviruses enters the cell. The RNA genome is then replicated and our mRNAs are transcribed. And in the process, it generates viral double-stranded RNA. And this is a general characteristic really of all RNA viruses, and some DNA viruses actually produce double-stranded RNA as well. And this is recognized by several sensors in the host cell. Most notably, MDA5 recognizes coronavirus viral double-stranded RNA and not the alternative RIG-I. MDA5 then signals through MAVs to produce type 1 and type 3 interferons, which then signal through the STATs to induce interferon-stimulated genes and cytokine responses downstream. So this is kind of the canonical interferon induction pathway. Double-stranded RNA is also recognized by oligoadenylate synthetases, shown here. They produce 2,5-A, which is a small oligoadenylate with a strange 2', 5' linkage, unusual, which then induces dimerization and activation of ribonuclease L, which cleaves host and viral single-stranded RNA, leading to cell death and inflammation. At the same time, PKR is also recognized double-stranded RNA. It autophosphorylates and then phosphorylates PEIF2 alpha, which is a translational initiation factor, leading to translational shutdown, cell death, and inflammation. So all of these pathways can be initiated in parallel, but interferon also upregulates further OASs and PKR and kind of superinduces these pathways. So we know now from past literature that NS2 and NS4B of both of OC43 and here of MERS encodes these 2', 5' phosphodiesterases that cleave this molecule, leading to complete shutdown of this pathway. NS4A of MERS is a double-strand RNA binding protein that binds to double-stranded RNA, kind of slowing down all of these pathways. And then ORF6A of SARS prevents nuclear translocation of STAT1 and slows down the induction of interferon-stimulated genes. And then all of these viruses encode NSP15 or ENDO-U that I mentioned earlier, which is one of the replicase encoded proteins that has an interesting function. It actually cleaves the genome RNA at some point, presumably to prevent the accumulation of more double-stranded RNA. So all of these proteins really act together to induce in various combinations in various viruses to slow down these pathways, these host response pathways. So we wanted to see, there was really very little known about the interaction of SARS-CoV-2 with these pathways. So we used a variety of cell types because we knew from our work with MHV that these responses can be very cell type dependent. So we used different cell types. We also used MERS as the kind of negative control for a virus that barely turns on any of these pathways. And we used Synbis virus as a positive control because we know it turns them on really well. So here we infected nasal epithelial cells derived from patients with SARS-CoV-2 and with MERS. And we can see that in the red that this is staining for viral nucleocapsid, that all the virus replicated well in 4 donors, cells from 4 donors, and MERS coronavirus also replicated. But when we looked at the kinetics of replication, we found that, and again, this is 4 different patients, that SARS replicated with much longer kinetics than MERS, shown here. And also to a higher degree. So we wonder whether this reflects the fact that SARS seems to hang out in the nose longer than MERS, and maybe helps with the transmission of pre-symptomatic patients, perhaps, speculation. And we can see here that there's a bit of interferon induction, but really not very much, shown here. Interferon beta, interferon lambda. We also looked at a couple of interferon stimulated genes, OAS2, IFIT1, and this is the MDA5 gene, and just one chemokine that's known to be important for innate response in the nose, and very little induction. We also curiously found very low levels of ACE2 by Western blot in these cultures, although we know that ACE2 is expressed. It's just not expressed at a very high level. We then looked at pluripotent stem cell derived alveolar type 2 cells. This is one of the main cell type infected in the lung. And here we can see that these cells are expressing T tomato from the surfactant protein C promoter, which is AT2 or alveolar type 2 specific protein. And we can see here that the cells are infected. This is staining for nucleocapsid and co-staining with the cell type marker. So these cells are also well infected. The virus replicates to pretty high titer, up to 6 logs of titer per mil of supernatant, which is pretty good infection. They induce not a whole lot of interferon. Here we compared interferon induction in SARS-CoV-2 infected cells and Synbis. Here's Synbis dwarfs the amount of interferon induced. However, there is some induction of interferon stimulated genes shown here by OAS2 and IFIT1, even better than Synbis. And we saw very little induction of RNase L that's shown here. We tracked RNA cell activation by looking at degradation of ribosomal RNA. So here in SYNBIS infected cells, we can see that ribosomal RNA is degraded to some extent, whereas we don't see that in the SARS infected cells. We then looked by Western blot at both phosphorylation of STAT, which is one of the steps in activation of interferon stimulated genes. We didn't see that. We did see pretty good PKR activation. Here's PIF2 alpha, which would suggest that protein synthesis will be shut down. Here's PKR itself, I mean phospho-PKR. We saw very little MDA5, which might have something to do with the low level of interferon induced. And we saw undetectable levels of ACE2, although we know that there is receptor expressed, only not very high. And we learned from our studies with MHV that coronaviruses don't necessarily need high level of their receptor to enter the cell. We then looked at again, iPSC-derived cardiomyocytes, given that cardiovascular symptoms can be part of coronavirus disease or SARS-CoV-2 disease. And again, the cells are expressing a cell type specific marker, C-troponin T in this case. And here's infected cells, this labeled with, stained with SARS-CoV-2 antibody against nucleocapsid. And here we see that we actually see cytopathic effect, which we didn't see in the respiratory derived cells. And we see cell-to-cell fusion in some of these cells, which is a characteristic of coronaviruses. The virus again replicates quite well up to 6 logs, again, as it did in the other cell types. And again, we see very poor interferon induction, interferon beta and lambda. We see more induction of interferon stimulated genes shown here. And again, and very little of these, well, these interferon stimulated genes barely induced. We see some here, but again, dwarfed by SYNBIS virus. We saw no induction of RNase L in this cell type. And then when we did Western blots, we again failed to see STAT phosphorylation, but we see again robust PKR and PIF2 alpha induction shown here, similar to SYNBIS. But we saw no expression of RNA cell, MDA5, or MAVs, which may express or explain some of the lack of induction of RNA cell or interferons. And this may be because we actually looked back and found this was already documented for this cell type, and this may be to prevent inflammation in heart muscle. Okay, so with that in mind, we then we compared the basal levels of interferon and interferon stimulated genes in these cell types, and we found interestingly that the nasal cells have really much higher basal levels of expression of most of these genes, particularly interferon lambda, OAS2, and that may make sense in the sense that nasal cells are exposed more to pathogens in the environment. And I think that's important because the higher the basal levels, the more easily that these responses can be induced by viruses. That's something we also learned from MHV's system. So, okay, so now we then looked at some cell lines where we could do a bit more mechanistic studies, and we found that SARS-CoV-2 infects CALU3 cells and A549 ACE2 expressing cells. Both of these are airway derived cell lines. This cell line did not express ACE2, so we transfected or we transduced it to express a pretty high level of ACE2. And Vero cells are the typical cell line that we make stocks of virus in. And we can see here that all 3 cells lines are well infected by SARS. This is again nucleocapsid staining. They all produce double-stranded RNA as expected, shown here. And here's the merge. And you can see here that there's a lot of syncytia formed or cell-to-cell fusion in the A549s and the CALU3 cells, which makes it difficult to see the double-strand RNA is localized in factories attached to the ER near the nucleus. You can't really tell that in these cell types because of the fusion. And the virus replicates quite efficiently in Vero cells and A549 cells, shown here. And also in the CALU3 cells, where we compared replication to MERS, shown here, MERS coronavirus, and a MERS mutant lacking its interferon antagonists. So SARS replicates less well than MERS, and even a little bit less well than the MERS mutant shown here. And we're going to compare, in some cases, SARS replication and SARS induction to MERS and this MERS mutant that already is lacking 2 known interferon antagonists. Okay, so this shows interferon induction in the A549 ACE2 cells. Again, really poor compared to Synvis. And then induction of interferon stimulating genes occurs only really late in infection. This is 48 hours post-infection, and really much less than MERS in some cases, but here really similar to Synvis. I'm sorry, Synvis. And here very little induction of IL-8. And when we looked at we also saw really undetectable STAT phosphorylation. But when we looked in the Calu-3 cells, we saw a slightly different situation. We saw induction of interferon here, interferon beta and lambda, much more so than MERS, which really shuts down the system, and more similar actually to the MERS mutant. And when we looked at interferon stimulated genes, this occurred really earlier than we saw in the other cell type, and much more so than with MERS, and even more so than the MERS mutant shown here. Again, this mutant is lacking interferon antagonists. So we learned from this that MERS behaves more, SARS-CoV-2 behaves more like MERS, which is already lacking antagonists. And then when we looked, we did see STAT phosphorylation in SARS-CoV-2 infected cells, as we did in MERS-CoV-2, the mutant, but not in MERS wild type. It's shown here. We then looked at OAS RNA cell induction and PKR pathways, and we saw again that SARS-CoV-2 activates RNA cell degradation, shown here, in both cell types, the A549s and in the Calu-3 cells. And here again, it behaves more like the MERS mutant, which activates some RNA cell, whereas MERS-CoV itself, wild type, does not. And here's just the Synvis control. So this is pretty good activation of RNA cell. We also saw activation of PKR by SARS-CoV-2, here shown here, and PIF2 alpha in the A549 cells, and again in the Calu-3 cells, where it's behaving more like the MERS mutant that activates PKR, whereas the MERS wild type really suppresses both of these pathways. And okay, and then the other thing we did was we produced knockout cells with knocking out either MAVs, RNase, or PKR from these A549 cells. And this just validates the knockout shown here. These are Western blots that we have cells knocked out for MAVs, RNA cell, and PKR. When we looked at replication in these cells, we found that in the absence of RNA cell, we had increased replication, whereas knocking out either MAVs, the interferon pathway, or PKR really had little effect on virus replication. When we looked at cytopathic effect shown here, this is just cells infected and stained for live cells. In the absence of RNA cell, we see a lot more destruction of the monolayer of cells, sort of supporting the idea that RNA cell really is restricting replication of SARS-CoV-2. And just in the last data slide, we found, we looked at activation of RNA cell in the absence of MAVs or PKR, and we can see here that SARS-CoV-2 and Synvis activate RNA cell perfectly well, even in the absence of the production of interferons, really showing us that RNA cell is an independent path from the interferon pathway and can be activated even when a virus can shut down the MAVs pathway. And we saw the same thing in the case of PKR. When PKR is absent, we still see activation of RNA cells shown here. And but we retain the phosphorylation of PIF2 alpha, suggesting that another pathway or other proteases, other kinases are shutting down protein synthesis, as shown here, even when we knock out PKR. So we're really interested now in understanding the downstream effects of activating or antagonizing these pathways, as we've seen with SARS-CoV-2. So in conclusion, coronaviruses enter cells by two pathways. The pathway used depends on spike protein protease cleavage sites and host surface and intracellular proteases. Coronaviruses express numerous conserved host antagonists in the replicase locus, often enzymes, and these are potential targets for antivirals that may be effective against most, if not all, coronaviruses. And the lineage A and lineage C beta coronaviruses express accessory proteins that contribute to antagonism of activation of RNA cell, interferons, and interferon stimulated genes, as well as PKR. And SARS-CoV-2, like MERS, weakly induces interferon. However, SARS lacks a phosphodiesterase and a double-strand RNA binding protein and activates RNA cell, promoting phosphorylation of PKR and STAT. These data suggest that SARS-CoV-2 is not as adept as MERS-CoV in shutting down double-strand induced antiviral pathways. And we speculate or question, could this contribute to SARS-CoV-2's asymptomatic infections and spread? I want to acknowledge my lab shown here. This was us before SARS-CoV-2. We really can't do this anymore. We're kind of shut inside or separated. And these are former members of my lab that are really important for some of the earlier work on MERS and MHV. And these are some of our collaborators and our funding.
Thanks, Susan, for an absolutely wonderful talk. This is a great kickstart to the TNQ Janelia series, giving us such a wonderful overview of coronavirus biology. So I'd like to bring in Shahid and Jeetu as well, so that we can kickstart our panel discussion. I'm going to take the advantage of being the moderator by asking the first one and then pull Jeetu and Shahid in for this discussion. You know, given you covered the entire spectrum of coronaviruses, starting with the history of it from the beginning, and that's fascinating. But what is it, given that SARS-CoV-1 and CoV-2 are so similar, what was it that was so different about these two that resulted in the first being contained at 8,000 and now this completely huge global pandemic?
That's clearly the biggest mystery, I think. That's the biggest question. And if you look at the genomes, for example, there's absolutely no clue why. I mean, we're starting to understand, I think we have to understand each one of these genes really well, but it could be something really subtle. Like we know a one amino acid change could inactivate an enzyme or something like that. So we really don't know. I mean, and there's not so much work on SARS-1 because, well, one reason is because it's a select agent. And for example, we stopped working on it because you had to get so much security that we just couldn't do it. So I think we don't know a lot of the we know a lot about SARS-1, but we don't know every detail about every gene in that virus. So we don't know in summary. It's really mysterious. I mean, MERS is even more lethal MERS is more lethal, but less contagious. SARS-1 is quite contagious. SARS-2 is quite contagious. So I think the reason it's maybe so contagious is it may stay in the nose longer before it goes down into the lung. But why it does that is really, I mean, there's one paper that says that SARS-2 replicates at a lower temperature better than SARS-1. So maybe that helps it stay up in the nose. But, but molecularly, there's really no clue to why.
Let's pull in Shahid and Jeetu. Shahid, you want to give us a little bit of understanding of the India scenario as far as COVID-19 is concerned? It'd be very nice to get your views and also your questions for Susan.
Sure. But before I do that, maybe a comment on the question that you just asked, Susan. I think one critical difference between the public health aspect of COV-1 and COV-2 is that COV-1 was shed primarily when somebody was symptomatic, whereas COV-2 has been shedding a few days before people become symptomatic. So there are a lot of pre-symptomatic people who don't know that they have the infection who are spreading it around. Another interesting thing is that eventually, the 2003 coronavirus was controlled using 19th century methods of isolating people, pure public health measures, despite being in the 21st century. That has not been possible here with the rapidity at which it has gone forward. You want to add something before I get to the India scenario?
No, I was just going to say, yeah, all that stuff is really important, but we have no clue as to why.
Right, exactly.
The virus makes it behave that way.
Yeah, so we don't know the mechanisms, right?
Yeah.
So looking at India, I was looking at the numbers today, and India has gone to 6.9 million cases. with 106,000 deaths. The outbreak in India appears to have slowed down in the last 3 weeks or so. So at peak around mid-September, India was putting out about 94,000 confirmed cases per day. Today it is down to about 75,000 per day. But to put this in perspective, India is still putting out about 24% of the daily global caseload. So that is important for us to understand when we say that our outbreak is going down. The second narrative that we constantly hear is the death rate in India is low. On a daily basis, India is still putting out 19% of the global death every day. So we are by no means slowing down. So this is something that people need to understand. If you look at the dateline of India's COVID situation, the first case was detected on 30th of January this year. And then from much of February, for most of March, we hardly had any cases. When our lockdown was put in on 25th of March, we had only 650 cases and 12 deaths. And by the time the lockdown ended, we had amplified the number of cases about 300 times and the number of deaths about 250 times. And in the unlock, you have seen that the virus has really spread exponentially. So today the situation is that 2/3 of the cases in India are coming from rural districts and villages. In some way, it is good in the sense that the outbreak will slow down because the population density in villages is not as high as in cities. On the other hand, the healthcare facilities in villages, especially testing facilities in villages, are poor. So it's a very mixed bag right now, and it's very, very hard to predict when all this will be over.
Let's pull Jeetu in. Jeetu, your views on the fact that this virus seems to toggle between two ways of getting into a cell, and viral entry being something that might of particular interest to you. So I pull you in on that for your comments.
I think Susan talked about how the virus decides which way it needs to go, given the fact that it can be modulated by the furin cleavage on production, and also requiring multiple cleavages to release its fusion protein from the spike glycoprotein that is its receptor. But the most important thing about the virus is that if it requires the intracellular machinery, it seems to require an extremely acidic compartment, and these acidic compartments could be very potent targets of many of the potential antivirals, and and interestingly, several drugs today. And this is something that we've been noticing as we do some work on this. Several drugs available today are drugs that inhibit the acidification machinery inside a cell. So if the virus is predisposed towards the endosomal route, and Susan didn't indicate that, and I'd actually be curious to hear what Susan has to say about what the SARS-CoV-2 is doing. In terms of its own entry process, whether it's using both or only one of its of these two pathways. But I think if it is using the endosomal route, then at least early entry inhibitors are a potential option. But Susan, I'm sure you have something more.
Yeah, I think from what we know, well, first of all, I think so the paradigm says for the AT2 cells that it's cleaved by timpers and goes through the surface, but I think it's much more complicated. I think that that may be true for that particular cell type, but other cell types may be more predisposed to the endosomal route, and that's why I think it's tricky, because certainly the endosomal route requires the low pH, and something like chloroquine or other low pH things that raise the pH in endosomes would theoretically be effective. But I think it's so hard to know, to depend on that pathway only. So if you shut down that pathway, I don't think it's sufficient in many cell types. And in fact, there was a really interesting paper that showed for, I forget which cells, but some airway cell that you needed really to inhibit both furin and TIMPers to shut down entry. So I think it's really slippery. It really depends on which cell type for which virus. And the other thing that's kind of curious is that SARS-1 doesn't have the furin site, the famously doesn't have it, and SARS-2 does have it, yet they're both invoked to use TIMPers and that pathway. So that's confusing to me. So.
Right. I think it needs double protease, cuts in order to be in order to generate this fusogenic peptide, and maybe that's where TIMPers is so important as well.
So just say from MHV2, it's really striking. This MHV that has zero cleavage when it comes out of the cell; it's completely uncleaved, and it seems to be much more dependent on the endosomal route, just experimentally. And also different cell types, even different cell lines that we used had different propensities to be endosomal versus surface. So I think it's pretty tricky.
You touched on this right at the very beginning of your talk to sort of discuss the entire conspiracy theory about this being a potential man-made virus, and you put that to rest. I was wondering your comments on what the natural reservoirs for this might be. There have been discussions about bat, of course, but pangolin and other discussions have come up. What are your views on this?
Okay, my views, just from reading informatics papers, which I have to admit I can't completely 100% understand, but there's some paper that shows really nicely, I think, that using molecular clocks and bioinformatics, that they can show or predict there was a precursor in bat that had this really good receptor binding site that would have a high affinity for the human receptor. And their study suggests that the pangolin viruses were derived from that. But at the same time, there was a recombination to produce this RATG13 is the bat virus that's most related. And this paper asserts that actually there was a precursor to all of these viruses in bats, and that we don't really need to invoke the pangolin. Like, it could even have gone from a bat to a human, or there could be an intermediate species. And I don't know that it even matters that much, but the point is that there are viruses in bats that could potentially infect humans, I think, whether or not may go through an intermediate species. But I think the pangolin's probably not the intermediate, in my opinion, from what I've read.
So do you think there's an amplifying reservoir somewhere that's not the bat, but something else?
It could be, but it also could be that a human got the bat virus, and then it somehow adapted to humans. I think the thing about it starting in the market is probably not correct from what I've read, that it might have started before that. That might have been an amplifying event, but yeah, so I think we don't know. And I think that's why I do think it's really important to keep looking for these viruses in different animal species, and not, that was sort of shut down by our government at one point. But it's important, I think.
So can I just add that the biggest substrate for viruses on this planet are humans, because humans have the largest population. So you don't always need an amplifier in animals. If the virus is adapted enough to transmit, it can very happily transmit from one human to another.
Really contagious, right? This one, because of what we talked about. Yeah, so it gets into a human and then it just goes.
Yeah.
Whereas, see, MERS, for example, MERS is not that contagious human to human, and it definitely has a reservoir. Like, it's really common in camels in the Middle East and in Africa. But a really strange thing about MERS is that it seems to only transmit in the Arabian Peninsula, that camels in Israel, for example, or in Africa, the people don't get it from them. It's a real weird mystery. So.
Vidita, a question for you about the SARS-CoV-2 variants or strains that have been seen within India, and also along the lines of the emerging mutations that are being seen across these strains to all three of you. I mean, comments on that.
Right. Well, maybe I'll take the question around the strains that have been seen in India, because we just completed a study on that. I mean, when I say we, a number of institutions of the DBT, led by the National Institute for Biomedical Genomics, Shomitro Das and colleagues from Bangalore as well. And I mean, what we find is that the original virus strains, and this is tracking viral genome sequencing for over 1,000 genomes, viral, and from late February to now, to July. And what we find is that, the initial influxes are, of course, coming from Europe, and the strains mirror precisely what is there. And then they acquire these more so-called more infectious variants, like the D to G mutation in the spike glycoprotein is supposed to be more infectious. And then, specific mutations, in outbreaks, for example, in Maharashtra, generate multi-site mutants that seem to be spreading rapidly, and seem to be, in fact, creating sort of a strain-dependent spread in some parts of India. So geographically, as well as in terms of the different mutations, there seems to be variants that are circulating in India, which are perhaps unique here, but that's not to say that the same thing is happening in other parts of the world where the virus is circulating with its own unique variants. And it's, unclear that there is anything distinctive about the mutations that cause any difference in the pathology or virulence from anywhere else in the country. So if that was also one of the points you were raising about differences of the virus strains in India, because that has been a point of discussion here, that by different virus strains may be circulating in India, therefore the death rates are lower. But I don't think that seems to be borne out by the evidence from sequencing the genomes from these consortia efforts.
I think coronaviruses don't change a lot in general. The proofreading enzyme is thought to be one reason, but I think the only predominant, the most famous mutation is the D to G mutation in the spike, but I don't think anyone's really proven that it has anything to do with pathogenesis, as you said. So my gut feeling is that there'll be lots of mutations, but that none of them are going to be really associated with more pathogenic outcomes. But I could be wrong, obviously, but I think that's generally true for coronaviruses, that they drift and they change, but not a whole lot. Where did the ones in India, where did they come from? Can they be traced to?
Yeah, so I think the earliest ones seem to be traced to either the Middle East or Europe. At least that's from the cases that we have. Maybe Shahid has some more input on that.
No, you're right. It's mainly from UAE and Europe. Some isolated ones initially from China, but not a whole lot from China, mainly from Europe and Middle East.
I'm going to take the advantage of the last question and target it towards Susan. First of all, thanks again for an absolutely wonderful overview and discussion on the biology of coronaviruses. So, big question, which many of us have, which is there going to be a SARS-CoV-3? And if so, what does the scientific community do to prepare itself for that?
I think there'll be another one, I don't know, but it may not be a SARS-CoV-3. It might be a MERS-CoV-2. It might be a, XX-CoV-1. So that's why I really stress the idea that antivirals are important. Clearly we need a vaccine now for this virus, but it might not work against COVID next, but a remdesivir should, or if we had a combination of drugs, at least in the short term, or this like plasma or this monoclonal antibody treatment that our president had recently, something like that. He's laughing, but that would be more specific to the virus. So I should take that back. But the drugs against the replicase proteins, I think, are really important that way, and also to be prepared in public health wise, right? To have the PPE and know when to shut down and what to do when. That I think some countries actually were much better at, like Japan, I think. And I mean, our country was terrible, really. So yeah.
Thanks, Susan. Thanks, Shahid. Thanks, Jeetu. We'll hand over to Janine and Sharda to take over for the open Q&A questions.
Great, perfect. Thank you so much. We do have some audience questions, so we will just start right in. The first one is a YouTube question for Susan. They want to know, under what conditions does the virus follow an endosomal pathway to enter the cell, and can that impact virulence and nature of the progression of the illness?
Can answer parts of that. It'll enter the cell that way, probably if it hasn't. It depends on the cell. Some cells may just not have the correct proteases on the surface, and it would sort of favor an endosomal pathway, or the virus itself may not have this cleaved spike, which in the case of SARS-2, we know that it does, but SARS-1 didn't. So that will sort of usher its way in. Combination of the cell type, the proteases available, the spike protein. Whether, I think it's hard to predict. I know that I've read now that a virus with an uncleaved spike in terms of SARS-2 is not pathogenic in hamsters. So in that case, it seems that it matters which way it goes in, but maybe it doesn't have to do with the pathway. But in terms of the mouse viruses, entry by either pathway leads to a pretty severe disease for the mouse. So it's kind of unpredictable, is the bottom line, I think. In terms of pathogenesis, it probably depends on the particular virus.
Interesting. So maybe that explains why different cell types show different levels of pathogenesis to this virus.
Yeah, I think so. I think so.
All right. So here's your next question, Susan, also from YouTube. Why does coronavirus have several frame shifts during replication? If so, what is the significance of this frame shifting?
Okay, so as I said, so the ORF1b proteins, including the replicase, are probably produced at about 20% the level of the ORF1a proteins. Why that happens, I don't know, but it seems to be a really common thing among many RNA viruses. They may not have frame shifts, but they may have other mechanisms to regulate the amount of, say, the polymerase versus the other proteins. And I've just noticed that it's like alpha viruses also produce lower levels of replicase. I think that's true for retroviruses too, that produce the replicase as a fusion protein. So for some reason, these RNA viruses don't produce, they sort of produce lower levels of replicase. So I think the reason the frame shift is there is one way to regulate the differential expression of one group of genes versus the other.
Thanks, Susan. Another question from our audience is, where have most observed mutations been clustered on the SARS-CoV-2 genome, and is that significant?
Ooh, I don't know the answer to that, and I don't think there have been. I don't think there's a place that they've been clustered. Although I did hear that I think in tissue culture they accumulate mutations near the cleavage site, but I don't think, other than this D to G mutation and maybe some of these ones that you were talking about accumulating in India, I don't really know of any generalization except to say that mutations happen, and maybe one of the other panelists knows better.
So, the thing about mutations is that mutations happen all the time, but you only get to see those mutations that provide any benefit to the virus. And clearly the D to G mutation is providing some benefit. The way this mutation has taken over, initially it was all D, and then very quickly it's, more than 2/3 G now. So it's clearly providing some advantage to the virus, and it's possibly because glycine very close to the cleavage site, as opposed to a more bulkier amino acid, provides better access to the cleavage site. That's one possibility. So mutations happen all the time, but they have to provide some benefit to the virus.
There's also, I've heard for the D to G mutation that the S1 protein is supposed to be more stably associated with S2. But what's strange to me about that is in the mouse virus, when S1 is less stably, it's the most virulent strains that have a less good association. In other words, it seems to be an advantage, at least in that system, for the S1 to come off easily and sort of trigger a fusion more readily. So it's kind of the opposite of what we see with the other virus. So I'm not sure that that particular factor is what makes it maybe a better, or what it seems to enter the cell more readily, at least in vitro assays. But I think all of this stuff needs to be done with we have reverse genetics now to take the mutation, put it into a wild type background, isolate that mutation and say, just what does that mutation alone do to entry or pathogenesis? Because it's really hard to say, because the D to G, I think, comes along with another mutation. I forget, in the polymerase, maybe there's often another mutation. So it's hard to really be sure that what it's doing, but it does seem to be selecting, right? It seems to be selected for, like you said.
So another question from our audience is a similar antiviral pathway observed in asymptomatic patients, since they don't seem to trigger that cascade for the manifestation of inflammatory symptoms?
That's a really good question. So it seems like these interferon pathways are shut down in patients early, but then later they're important to get an interferon response, or no, if they're shut down early. So if they're treated with interferon, at least this is true from animal models, that it might shut down, prevent the later disease, which sort of makes sense, like using an antiviral. But then later in infection, this sort of dysregulated cytokines, including interferon, is pathogenic. So why that happens, we don't know, although there have been a couple of studies recently that showed some genetics of people that have this really, dysregulated interferon response. It may actually be a genetic thing that predisposes to the inflammatory, the really bad inflammation, but I think we don't know enough about it yet.
Thank you, Susan. Thank you, Shahid. So switching gears back to genome and mutations, you mentioned a furin cleavage site and how SARS Classic does not have one, while SARS-CoV-2 has one. So how did this furin cleavage site originate? Is it recombination, mutation? What's the hypothesis?
That's the one thing that I think is still really mysterious. So the paper that I was referring to that showed sort of the predicted precursor viruses, that's the one thing they don't talk about in that paper, is where the furin site came from. But certainly there are a lot of coronaviruses that have furin sites. It's really not unusual. Like all the lineage A viruses have it, and all the lineage C viruses have it. So MERS has one, OC43 has one. So we don't really know. I mean, it could have happened by a recombination event, but I don't know. And that's I think that's one of the suspicious things that the conspiracy theorists talk about, like that it has this site, but I don't think it means a lot.
Another question that we have from the audience, which I think is something that we've probably all wondered at one time or another, is how do bats successfully deal with coronaviruses? And is there something that we can learn from that?
Well, there's there's theories about that. One of the theories is that they have really high innate immune responses. You know how I talked about basal level of those interferon genes, that supposedly bats have really high interferon alpha, at least some bats. So that's one of the ideas, that they kind of coexist by kind of being more immune to them. I don't know whether that's true. The one thing we did in bat cells is we looked at that RNA cell pathway, which is something that my lab we were really working on that pathway before the virus came. Anyway, and in bats, that's very similar to in humans. So, that's probably part of it, but a lot of people are working on that.
Thank you, Susan. Here's a question for Shahid. Should India be thought of as one entity in projecting the disease spread, Or should we break down India into different regions and coming up with a better understanding and better forecast for the future?
No, no, obviously India is a big country and conditions differ. So you can't compare the city of Mumbai to Arunachal Pradesh. I mean, population density differs, host genetic differs, a lot of differences. So yes, India has to be seen as, and if you look at India today, there are multiple outbreaks happening in India. Some are going down, other regions are going up, other regions are more stable. But if you want to really model, the critical thing that you need for modeling is good granular data, which is often not available, especially at the state level. So it's very hard to say at this time when you look at the numbers, what the numbers truly mean. But to the original question, yes, I mean, India can't be seen as one monolithic entity. It has to, there are multiple conditions in, different regions.
We have a question from our YouTube audience directed to Jitu. For viruses that enter through the endocytic pathway, in addition to cathepsin cleavage, can lysosomes degrade the viral nuclear material in order to stop the infection?
Well, that's an interesting question, but I mean, moment the virus gets into these endosomes and the spike protein gets revealed in its fusogenic capacity, it's likely to just pop into the cell. So, unless one prevents that, it's not going, we won't know. But it's clear that, if you inhibit the virus, which let's say the MERS virus, which can only use the endosomal route, the virus does not replicate. So and whether the inhibition itself leads to a clearance of the virus on its own because of the lysosomal degradation capacities, I think that still needs to be determined. But the virus certainly doesn't replicate when you prevent its entry into the cell. And it's probably getting degraded. But here again, I think detailed studies would be needed, and if Susan has some more evidence of that, she might want to say something.
No, I think we don't know. It's too hard. It's too hard to find the virus in the endosome. I mean, it's even been really hard to find the second cleavage product. Like everybody can, it's easy to see when the spike is cleaved down the middle, but the second cleavage site produces this small S2 peptide that people don't find that easily. So it's hard to know those intermediate steps.
Thank you, Jitu and Susan. Switching gears is a question related to immune response. So generally, what is the mechanism? You talked about the viruses shutting down interferon response. So what is the mechanism of the shutdown? And a related question, why is MERS-CoV-2 more efficient at shutting down interferon response?
Okay, so I think the reason that MERS is lacking at least the two proteins that we've studied a lot in our lab, the phosphodiesterases that shut down RNA cell, and the NS4A protein, the RNA binding protein. So it's interesting because they seem to need multiple proteins to really shut things down, like that endo-U that I mentioned, endonuclease U. All the viruses have that, yet it does prevent or reduce the amount of double-stranded RNA that accumulates, but they seem to need also an accessory protein to really shut down. So we think that SARS just isn't as efficient, it doesn't have these proteins, so it allows more production. I would guess that SARS-1 is similar, but I'm not really sure. So SARS-1 has this ORF6 protein that prevents interferon stimulated gene an induction because it prevents the STAT proteins from getting into the nucleus, and those are needed for transcription of the interferon stimulated gene. So we don't know if that's true also for STAT2. That's one of the things that one of my grad students is trying to work on now. So in short, I think that it's lacking some of the interferon antagonists that some of the other coronaviruses have. Why it does that, I don't know, but maybe that's why it's less virulent. I mean, it does, it's horrible and it spreads like crazy, but it's actually kills a lower percentage of people than either MERS or SARS-1.
Thank you, Susan. And I'll hand over to Janine now.
Thank you, Sarada. I think we're just about out of time today. I know that we did not get to all of the questions. There are so many, but I think we do need to let Susan and the other panelists go and move about the rest of their days. So as we do that, I just want to say one last big thank you so much to our speaker today, Professor Susan Weiss. We really enjoyed having you and we learned a lot. I also want to thank our panelists, Jitu, Mayor, Shahid, Jameel, and Vidita, Vaidya. It was really a pleasure listening to your expert discussion. I want to thank our audience for your really great questions. I am sorry that we could not get to all of them, but I do encourage you to join us next week for the second seminar in this series. It's October 16th. And the speakers are Joseph Derisi on the CLIA Hub, student volunteer run COVID testing, and also Drs. Ulas Kolthor and Sandeep Juneja, who are going to talk to us about the Mumbai Sero Survey, facts and interpretations. So I hope to see all of you back here again at the same time next week. Bye-bye, everyone.